The cerebral, also known as the brain, sends an electrical current to a neuron. In this part of the video, the table will represent the neuron. Then action potential carried by the motor neuron meets the muscle at the neuromuscular juncture. Acetylcholine, a type of neurotransmitter, is released between the sarcolemma and the axon terminal of the muscle fiber. Sarcolemma receptors then bind to the acetylcholine. Sodium ions then move through the membrane once the sarcolemma ion channels open. This process creates a muscle action potential.
Through the T tubules, the muscle action potential moves along the membrane. Once the muscle action potential is generated, an enzyme named acetylcholinesterase breaks down the acetylcholine. This makes sure that only one muscle action potential occurs from one nerve action potential. Calcium ions are then released from the sarcoplasmic reticulum. Then the released calcium ions then begin to flood into the sarcoplasm. Myosin binding sites are then exposed once the calcium ions bind to the troponin on the actin filaments. Myosin heads contain ATPase, which splits ATP and releases energy.
With the aid of tropomyosin, the protein, the myosin heads connect to the myosin binding sites on the actin. The working stroke is produced when the myosin-actin cross-bridge is turned to the center of the sacrament. Once that occurs, ATP will then bind to the myosin head, which leads to the disconnection of myosin from the actin. In the event where there is no longer any action potentials, the amount of calcium ions in the sarcoplasm decreases. After that, the troponin-tropomyosin complex moves to the initial location it started from, meaning the myosin binding sites are blocked, causing the muscle to relax.